Welcome to this session. I'm Dr. Mohit Mazumdar, and I have a PhD in computational biology, and I'm an expert in computational structural biology, and have several publications to which I've worked throughout my research and industry career, helping some research groups and during also my PhD work. So today we are meeting to discuss and to learn about some of the aspects of structure prediction. So as we all know, and it would be great if all of you can also introduce yourself in the chat, and we can have maybe a better interactive session here. As I'm also requesting that if you also have a protein structure that you want to predict in your mind, and if you can share the link, we can perhaps take a look at that also in the session. So it's an opportunity for those who are actually doing something related or would want to learn about protein structure prediction and how to use the AlphaFold, and more importantly, what next after that, like if how to validate what have you predicted, is this making sense on or not. So please feel free to share those information. You can send it to me using direct message, the protein sequence of your interest, and we can take a look at that during the today's session. And we'll try to be we'll try to do this together. And if you have those softwares installed, it would be great to do this in sync, which would be to have at least Chimera installed. So it's a visualization software, UCSF Chimera. So if you're on Mac, then you can use the X version, Chimera X, which also has this capability to predict structure from the interface, which is incorporated in the Macintosh version, Mac version. And the one that we have on Windows, which we will also take a look. So I have both the systems, Mac and Windows. Today I'm using two logins. So in case you face any difficulties and you have any questions, feel free to ask independent of the platform. Now about the session. So AlphaFold. And AlphaFold, I think we've been discussing this for a while, and also we have this ongoing training program going on, and I'm hoping that some of the participants from the program whom we are working with throughout the sessions and mentorship have also joined today's session, and we look forward to their questions. And in today's session, after this wonderful introduction, we have the demonstration and some of the concepts that I wanted to discuss about, some of the basis of what we are doing and what is different. And then we have also a guest lecturer, Dr. Puneet. I'm hoping that he'll be able to join. He's kind of busy, but he said that he'll be able to join and answer some of the questions that our participants have going through his sessions during the program. All right, so let's start with that. And again, yes, Sarah. Hi, Sarah. Yeah, we can try with your sequence. It's just that there's one limitation with sequences. Again, I should tell you this, that for predicting sequences of, which are larger in size, in length, the number of amino acids they hold is kind of directly proportional to the time that it is going to take. So usually the predictions run on GPU and CPU. Still, because there are a lot of people using this, so the prediction would take an hour and sometimes more. So we will go through that entire process. And yes, Sarah, we will have that. So I have worked on this on the project, and we will schedule a meeting maybe tomorrow or day after to discuss your project and what are the other the next things, because I think it is mostly about mutations, and mutations are so important in the current era where we're looking at the translational aspect by looking at genomic data information, and we're looking at the mutations and what are the implications in the structure. So AlphaFold is one of the way to look at one of the snapshots, but obviously today we are going to discuss that what is actually required to be able to make sense of such a project where you're trying to understand what would be the implication of the mutation on the structure, which could be a single mutation, which usually will not cause any change, major change in the structure if you're modeling it and just replacing one residue with another. But in folding and in functional implication, how that change in residue can cause the change in interaction network of the entire protein and have a devastating effect or some new features. So yeah, all right. So as you know, AlphaFold 2, and I am hoping that you all are aware of what AlphaFold is, because we also have covered it at one of our free webinars. If you have not, then you can ask the team to share that video with you, where we have also shared a video by the team by AlphaFold. So AlphaFold 2, AF2, was the star of CASP14. And what is CASP14? It's the biannual structure prediction experiment. So what they use is that they used novel deep learning, and AF2 predicted the structure of many difficult protein targets at a near experimental resolution. So I hope that you understand resolution. When I say resolution, it means that at how much detail we got from those experiments, and what level of accuracy in in the context of AlphaFold prediction is the closest to the native structure. And we do that by comparing structures. And today we are going to learn about that as well, that how do we compare structure, what is the statistical value that is important to understand, that how 90% similar and how 80% similar, what is root mean square deviation, all of that. So now coming back to the point where what I was trying to say is that that how will the availability now with AlphaFold and other softwares, you have this option to predict structure of the proteins, and they are at a decent accuracy, and they say that it's close to the near experimental resolution. So now there's an availability of high resolution structures, how it is going to impact biology. So first, structure alone does not provide the biochemical function of the protein, nor can AlphaFold 2 predict the phenotypical function, which is often the result of complex interplay of proteins. So there could be RNAs, there could be DNAs, there could be metabolites, which could give us the complete picture. Now, that's why one of the key steps for doing this is the first step, which is to get the primary structure, and then from that three-dimensional structure, you try to model protein interactions by looking at the complex structures and the complex interplay of protein and DNA and all of that. So that is one of the possibility to be able to do that model further, like go one step ahead with this predictions. So another thing is that the structure of a protein can use to infer its biochemical function, right? That's one of the objective. But you can also perform virtual ligand screening to predict which small molecules or drugs it might be binding and how it is binding. So that is again a session that is covered in the last session, where we covered virtual screening and softwares involved in virtual screening, which are publicly available. Anyone can start using them, download the publicly available data from ChemIMBL and other repositories, and look for similar structures, and use cheminformatics, and then use virtual screening softwares, and find out maybe top 1,000 or top 100 molecules which you would want to go next into with them, using maybe docking or other protocols. Now that's the strategy usually for such research. Now sorry for maybe the image below, it's not coming high resolution, but what I wanted to say is that even though AlphaFold 2 can accurately predict the backbone and the side chain conformation. So here on the screen, you can see that the typical conformation with all the atoms which are forming the peptide backbone and different side chain conformations. So AlphaFold is kind of predicting the backbone and the side chain conformation, and it is for a particular conformational state. Why it is for a particular conformational state? Because when you crystallize proteins, and this is based on the available experimental structures, that protein is another snapshot of a conformational state. However, a protein can exist in more than one conformation. It can have one conformation in an inactive state, and it can have another conformation in an active state. So if you have an active conformation, if an active conformation is predicted by AlphaFold 2, and then using it to target the inactive state, it will not work, right? Because there is a slight differences in the conformation. And below the image kind of shows different conformation of a maybe a loop region in a protein, and that itself can change the function of the protein and the interface. And having that multiple conformation allows ligands to bind, or specificity, which is very important in drug discovery and drug designing. So prediction of protein structures via the representation using backbone dihedral angles that you can see on the screen has really recently achieved significant progress. And that's why I was kind of discussing in this slide is that what we have been used to is structure based predictions, and those predictions are based on multiple sequence alignments, and then finding similar structures, and then modeling using them as a template. And that has been a common practice using either modeler, Swiss model, and all of that, which works really well for closely related proteins. Now, the recent achievements, and this is where everyone should know, is on the ongoing surge of deep neural network. So the deep neural network in research in general has a huge implications and has been used so widely. So today we are going to review some of those aspects of how the advancements have come up in deep neural network. And and also we observed that in the protein backbone angle prediction research, there has been an overall trend to employ more and more complex neural networks. So instead, like what people are doing, they are adding more features. Many of these research groups, they are adding more features that might add to more predictive power to the neural network. But then there is this important aspect in machine learning, is that when you have, it could be argued that when you have redundant features, that could rather clutter the scenario, because more complex neural networks needed to be counterbalanced with the noise. So, and this is a practical experience that people from machine learning has maybe experienced, that when we use a lot of features, then there's a lot of noise that has to be countered. So from artificial intelligence and machine learning perspectives, problem representation and solution approach do mutually interact and does affect performance. Now, that's the key takeaway. Now, a case study. Okay, Shruti and Oluwastosin, thank you for sharing about your current degree and your interest. Now, as we are discussing this as a understanding that how the new structure prediction softwares are being used, how they work. So before we go into the live session, the demo session, the objective is for you to understand that what is the logic, how it works, what are the advancements. So that's why we are covering this, and I hope that this is informational. Now, something new. So on July 2022, a Chinese group, biotech firm called Helixon, they launched this software called OmicsLogic. I don't know if you have heard about OmicsLogic or not. This is after AlphaFold. So the first computational method to predict, they claim it to be the first computational method to predict high resolution protein structures from a single primary sequence also what AlphaFold does, but this is using a different algorithm. So this new study by this Chinese researchers fills a much encountered gap in structure prediction and inches closer to the protein folding in nature. So OmegaFold is a protein structure prediction algorithm that uses a protein language model to predict structure. The algorithm works without multiple sequence alignments, which means it's faster because AlphaFold and also works on divergent sequences compared to AlphaFold because it uses multiple sequence alignment. And one of the reasons, if you compare, so unlike AlphaFold, OmegaFold does not rely on the MSAs, and then OmegaFold potentially better suited for proteins that have low sequence coverage. Uh, because multiple sequence alignment when you do may help OmegaFold, then because it has that additional information, but it could work without it as well. So OmegaFold can predict up to at least almost 4,000 residues and all. Then about the comparison and the understanding that what are the different models that are available that are really at par on CASP, the recent CASP, where there was a competition and people are checking accuracy. So the new model developed by Helixon, they claim to be outperform Rosetta Fold. So another important software in structure prediction is Rosetta Fold that is widely used and achieve similar prediction accuracy to AlphaFold 2 on the recently released structure. So in a study, the researchers said that they have used a new combination of protein language models that allows them to make prediction from a single residue. And this is by using geometry-inspired transformer model trained on the protein structure. So OmegaFold leverages a deep transformer-based protein language model that is trained on large collection of unaligned and unlabeled protein sequences to learn single and pairwise representation and use it as a feature to model the distribution of sequences. Now, the Omega protein language model, which they are calling PLM, can capture using that. What this does, like in a layman's term, is that it captures structural and functional information encoded in the amino acid sequences through embedding, and then they later feed it to a geometric inspired transformer neural network to distill the structural and physical pairwise relationship between amino acids. Finally, they predict out of all of this. This is kind of complex, but when you think about it the features, then it is actually not, and it's developed by research groups. So our objective is not to do this and develop something, but to utilize it and understand that how they are different, which tool to use when. So this is a very important aspect of bioinformatics, because thousands of tools are available online, which one to use for your research, and this varies from problem to problem. So that's why biology is kind of challenging. So again, that's what I was coming at, that all of this, then finally they do a structural module that predicts the 3D conformation of all the heavy atoms. Another folding algorithm is called the ESM fold. It uses a large scale language model for protein prediction. The improvements in the language modeling, perplexity, and the structural learning continues through. They have 15 billion parameters. Just imagine that. It's just because of the high throughput and this now computational power. This has been possible through deep neural network, and that's what I was putting emphasis on comparing few algorithms which uses deep neural network, and AlphaFold uses a neural network based architecture, and the training proceeds based on evolutionary, physical, and geometric constraints of the protein structure. So that's AlphaFold, and then you learn about OmegaFold, and then ESM fold, and then Rosetta Fold. So all of these folding softwares, they are kind of comparable, and they could be used to, if you're working on a really important problem, you would want to take opinion. Like when you visit a doctor, you have something serious, you want to take opinion from several of the doctors. Similarly, in this case, when you are trying to predict a structure, you would maybe take a look at the other predictions and then evaluate which is the best. Now, this is the overview. This was the overview. This was the understanding of how we can maybe use multiple softwares, how they work, understand, and then use it for our own problem.